All right, we're back with the light reactions of photosynthesis. Um, so the light reactions are going to be mainly kind of focused in and around the thylakoid. All right, the thylakoid is going to take light energy and it's going to convert it into chemical energy. Just a refresher on light. Light's electromagnetic energy. We tell the difference between types of electromagnetic energy by their wavelength. If you look here, we have a spectrum, the electromagnetic spectrum. Tiny waves, we have gamma rays. They're all the way on the left. Large ones are radio waves. They're all the way on the right. They can be like a kilometer long. So if you have a small wavelength, you end up having a lot of energy. If you have a large wavelength, you end up being like weak. Of course, visible light is kind of right in the middle of all this. Blue light tends to have more energy. Red light has less. And a side note, like your gamma rays and your X rays and UV light, those all have a lot of energy, relatively speaking, and those actually have enough energy. We call it ionizing energy, where they can actually, if they hit the DNA of an organism, they can potentially mutate it or change it. Versus stuff on the far right, like radio waves and microwaves, those are not ionizing energy. If your DNA gets hit with radio waves, it's going to be just fine. Interestingly enough, a lot of people are concerned about microwaves. My microwave oven is going to give me cancer if I stand in front of it, or radio waves like being transmitted to and from my cell phone could give me brain cancer. Well, I can tell you, the waves from your microwave oven and the waves from your phone, they're weak, relatively speaking. They're not going to be able to change your DNA and give you cancer. So anyway, pigments, things like chlorophyll, carotene, they absorb light. Okay, and then once they absorb that light, that energy gets transferred to something. If it's in what we call a photosystem. This is showing light entering like a chloroplast, hits a thylakoid. Some of that light gets reflected. Reflected light is not utilized by the plant for photosynthesis. Here's a three graphs or two graphs, and a top graph shows us a couple different pigments: chlorophyll A, chlorophyll B, and then something called carotene. All right, these pigments absorb blue light really well. They absorb red light really well, but they don't absorb green light very well. Likewise, if you look at the bottom, in the 1880s, there's a scientist by the name of Engelmann. He did this experiment where he took algae and then shined light on it through a prism. So that prism split the light up into kind of a rainbow spectrum of light. He added aerobic bacteria, so basically bacteria that need oxygen to like survive. What he's found is that the aerobic bacteria grew really well, kind of in the area where there was blue light, and in the area where there was red light, but not very well in the area that was green light. And that's because what we know now is, he knew this too, that when you have a lot of photosynthesis, you get a lot of oxygen production. So that bacteria grows well where the oxygen is at. The oxygen is by the blue, by the red, and not by the green. And so even in the 1880s, Engelmann figured out that green light is not very good for photosynthesis, but blue and red is. Okay. And this is just a quick summary of that. Chlorophyll A is kind of our main photosynthetic pigment, but we do have chlorophyll B and then some other ones like the carotenoids and something called xanthophyll. Like you see the carotenoids in leaves and xanthophyll in leaves during the fall, because that's when the chlorophyll A and B kind of degrades, and then you're left with those other pigments. So they're always there throughout the year when the leaves, but then chlorophyll A and B degrade, and then the leaves quote unquote change, but they only change because the green stuff goes away. So here's a picture of chlorophyll. All right, it's got almost like it's like a hydrocarbon tail, which allows it to embed into a membrane. The top part has got magnesium, which is an ion. It also has carbonyl groups that are going to allow it to be more hydrophilic. So here's a photosystem, and with a photosystem, light kind of hits it. That's that photon part, and then the energy gets absorbed, and then it gets moved to a pair of electrons. And those electrons, kind of hold on to energy, and then that energy is going to be used for stuff, which you're going to see here. So here we have a thylakoid membrane. All right. If you look on the left where it says photosystem 2, you can see light hitting the hitting photosystem 2. All right. That energy goes to the electrons. They get excited and they go through an electron transport chain, just like in the mitochondria. And just like in the mitochondria, hydrogen ions are going to get pumped. They end up getting pumped into the interior of the thylakoid. All right, so that creates a concentration gradient, kind of like with the mitochondria. So you're going to get a big buildup of hydrogen ions in the middle of the thylakoid. And then they're going to diffuse through ATPase or ATP synthase. Okay, and as they're doing that, they're going to make ATP. But if you look here, there we go. So the electrons keep moving on to another photosystem. All right, so we started out with photosystem 2, and then we're going to photosystem 1. Photosystem 1 was found first, then they figured out there was a photosystem, another photosystem, that's photosystem 2. Anyway, so light hits photosystem 1, and those electrons get excited. And then they go to something called NADP reductase. So remember, a reduction, it means you're gaining electrons. So those electrons go to NADP reductase, which get combined with NADP plus and H to make NADPH. So NADPH is holding on to kind of these high energy electrons. And the ATP and the NADPH are going to go to the dark reactions or the Calvin cycle. All right. The light reaction is almost done, except we have to take water here and replace these electrons that were here originally. All right, so they left photosystem 2 and they went to photosystem 1, NADPH. Well, we have to get those back somehow. Well, how do we do that? Well, there's a little enzyme here that splits water apart and turns it into oxygen and hydrogen ions, but it grabs these electrons and replaces the original electrons that were lost. And that, more or less, is the light reactions.